Hi, this is a video to help Bio24 students prepare for the tissues portion of the lab course. I'm shooting this video because of Hurricane Harvey. We lost a week of labs, but if you didn't just get hit by Hurricane Harvey, that's because I decided to reuse this video. Maybe it'll be useful. Who knows? So I'm Professor Alejandro Enriquez, for those of you who don't know me, and I'm going to go through a PowerPoint which is entitled 24 for Lab-Tissues, which is covers the material that you'll be expected to know for your first lab exam on the tissues of the human body. Now, briefly, first thing I want to talk about is the tissues are arrangements of cells, and I want to actually draw attention to the fact that you have cells. There's a cell. There's another cell, and these cells may or may not be attached to each other directly. So, for example, I may have cells that are attached to each other with tight junctions or other types of connective structures between the membranes of the cells, or they may be separated and may be attached to other materials that are around the outside of the cells. And these other material here is outside the cells. It's called the extracellular matrix. And you can really think of all tissues as having some tissues have an extracellular matrix more than others, and that's kind of understanding that is the key to understanding the difference between the different kinds of tissues. I'm going to go ahead and talk first about epithelial tissues. Epithelial tissues always come under this category. They do not have a well-developed extracellular matrix. Instead, they are basically all the cells are attached to each other, and we categorize epithelial tissues by the arrangement of the cells and by the shape of the cells. I can have cells in one layer, or I can have them in multiple layers. So that's one way we categorize cells. We call this simple, and we call this stratified. People will often start trying to use these terms, simple and stratified and so forth, to apply to other tissues, and that's automatically wrong. You automatically know you're wrong because we only ever describe a tissue as simple or stratified if it's an epithelial tissue, meaning it has many cells that these are all attached to each other with some type of tight junctions or gap junctions or other or desmosomes or other types of materials that attach the cells to each other. So we also categorize epithelial tissues only by the shape of the cells rather than by what's in the extracellular matrix, because there really isn't an extracellular matrix. So if in this case we would call this cuboidal, this would be simple cuboidal. You might think, well, this might be stratified cuboidal, but it's actually stratified squamous because we go, if this is the basal layer, the basal layer there, then we don't go based on the bottom, the cells on the bottom, we go by the cells based at the top, to the apical layer. So that's why this would actually be stratified squamous. If I had stratified cuboidal, even the top cells would be square or cube shaped. Or this might be stratified cuboidal here. And then columnar just means that the cells are very taller, right? So this would be like simple columnar here, right? This would be simple squamous. These are, note that we have a basal layer here, basement membrane at the bottom. So, and this is a image which looks like it's been cut off very slightly on your video. I apologize. Showing the different types of tissues and where they are found, different types of epithelial tissues. Now, one thing I want to draw your attention to, the function of epithelial tissues. Epithelial tissues are always forming a boundary of some kind. They always form a lining. That's what their purpose is, to be a lining. And the reason for that is that because cells are very good at controlling what goes in and out through their cell membranes. Remember, cell membranes are what we call semi-permeable. So if I have cells one after the next here versus if I have cells like this, only this arrangement would actually permit some things to pass through and other things not to pass through. I can allow some materials to diffuse through or actively transport, but I can block other things from going through. Here, there's no way to stop stuff from passing through to one side to the other. So this is why we have this as a lining. And all epithelial tissues form some kind of lining. The thicker the lining, the more it's meant for protection. Like on the skin is a very thick lining, stratified squamous. Obviously very thin linings like what you have on your lungs, or and also glandular or tissues are often thin so that they can secrete materials into your blood or onto ducts. Those are all going to be epithelial. This should never be epithelial. The extracellular matrix is never good at blocking things or good at allowing some things to go through it and preventing other things from going through. So that's what you see connective tissues normally look like that. So I'm going to briefly talk about the different types of tissues you're expected to know. First one up is simple squamous. So you can see that it's one very thin layer of cells. That very thin layer of cells are very squished down. This is actually the alveoli of your lungs right here. And sometimes you'll also see another example is the glomeruli of the kidney, where it's this thin lining around something in the middle. This part here is a simple squamous. Very thin. The next one up is going to also be a single layer of cells. This is actually simple cuboidal. What you're seeing here, take a look. There's each dark purple is a nucleus, and the nuclei are arranged in a ring. Some people mistake this for something that's stratified sometimes because there's just so much stuff going on they don't know what they're looking at. But you can clearly see that there's creating an environment, a difference in environment, the inside to the outside. What these are, in simple cuboidal case, these are tubules in the kidney that are creating a lining for the fluid inside that's ultimately going to become urine that's going out of the body. And so that's why this would be an example of simple cuboidal. It's one layer of cells inside to out, just like all epithelial tissues are going to have an inside to an outside. Another good example of inside to outside here, I have your simple columnar. This is in your intestines. Though we want to have one layer of cells so we can absorb things easily through from one side to the other, but prevent other things from going through. But we were absorbing things, but we don't want to absorb over and over and over. We don't want many layers of cells. So we just have one tall layer of cells here. And that's why these are simple columnar, and then the tissue folds. That's why there's another one right here. They're not stacked on each other. This is not stratified. Stratified squamous. Notice the basement membrane here is the bottom, and at the very top there's empty space. That's always your hint for looking at epithelial tissue. I know I'm looking at something that's going from one environment here. This is below the basement membrane of the stratified squamous. This is some other material here. And then, as you can see, the layers of cells stacked up one, starting here at the bottom, stacking up, and then they get thinner and flatter, and here at the very top they're flattened down. That's why this is stratified squamous and not stratified cuboidal. Pseudostratified is in reference to the fact that the cells look like they're stratified because the nuclei are at different heights, but if you look very carefully, you see that there's actually only one layer of cells. It's a constant thickness is your best hint. It's a relatively constant thickness. The cells are just kind of uneven in where their nuclei are. So this is the one you commonly see the lining of the trachea and your respiratory passages. The last one is a little tricky because it's transitional epithelium, which I guess we can think of as stratified cuboidal, but the trick with them is transitional epithelium is found in the bladder. And the bladder, as you may know, is very stretchy, and so when it's very distended, the cells are stretched out and they become more squamous, and when it's relaxed, they become more cuboidal or even columnar, which is why we can think of it as stratified cuboidal, but really we just refer to it as transitional epithelium. You have to be familiar with both the distended and the relaxed. The relaxed is on the left, the distended, fully stretched out, is on the right. And this PowerPoint you should have access to, so that's why I'm going through it a little quickly. You're just going to have to study it on your own. And again, a reminder, if you're not familiar, Dr. Bell's histology tutorials are also available. You can make your own flashcards and find histology quiz. I should have a Quizlet posted also from the internet that's good for testing yourself on your histology. So epithelial tissues, they're distinguished from connective tissues in that they're all cells, basically, that cells perform for creating a lining, allowing some things to go through and other things not to go through. That's what cells are good at. But of course, cells are mostly water. They don't provide any strength or toughness, and so that extracellular matrix, whatever might be in there, is going to provide the strength, elasticity, flexibility of your connective tissues. And so the different connective tissues, this is adipose tissue. This is actually the exception. This is the only one of the connective tissues that doesn't have an extracellular matrix. You can see the cells are kind of attached together, but they're full of that fat vacuole. That's why it looks like it's clear. This is a giant fat vacuole. The nuclei are squeezed into little bitty corners of these cells. So the connective tissues are united by having a similar function of providing a material strength or flexibility, in this case, of padding provided by adipose tissue. Your adipose tissue protects many of your vital organs and so forth. Blood is actually also connective tissue. Most of these guys are red blood cells. So white blood cells are the big white and purple guys, and the little itty bitty guys are platelets. Blood is also connective tissue, but note in this case we have an extracellular matrix that's liquid. It's water, basically. It's plasma. And so that would be why it's united under connective tissues. So now that we've kind of got the exceptions out of the way, the other connective tissues we cover in this class are, this is hyaline cartilage. This is a chondrocyte, and the chondrocytes are surrounded by this purple kind of gelatinous sort of extracellular matrix, which is a lot of protein and a lot of carbohydrate to kind of help provide a glue ground substance. So this is, it provides that, so the cartilage liquids in your nose and on the ends of your joints provides a kind of a sturdy toughness. But there's no blood vessels, so it heals very slowly. If you've ever broken your nose, you would know what I'm referring to. It doesn't go back very well. Compact bone, by contrast, it has a lot of blood flow. Now, people often look at this and they think each of these things is a big old cell, but actually this is what's called an osteon. It's an arrangement of many little cells. These are osteocytes. These are inside this matrix. And around it are the lamellae, which is the extracellular matrix of calcium phosphate crystals and protein and carbohydrate, collagen. So all of that makes this hard, solid material. Then in the very middle, this central canal here is a blood vessel that helps provide nourishment to these cells and helps regenerate the bone as it wears down so that your bones stay healthy and strong all the time, as long as you're eating enough food so that your bones stay healthy. Loose areolar connective tissue, these cells are fibroblasts. They produce the fibers that you can see, which are present in that connective tissue. And you can see it's all disorganized. It doesn't have a grain. That's part of why it's also called irregular sometimes. We contrast that with regular connective tissue. This is dense, fibrous connective tissue that's regular, where the fibers are all going in the same direction. The reason it's a little wavy is because the fibers are relaxed. Like there's an elasticity to them. It's like a rubber band. You pull it and it'll go straight, but if you kind of let it relax, it'll have a little bit of a curve to it. And so that's why that's why there's that kind of wavy look to it. So there's 4 tissue types. So 2 of them are the connective and epithelial tissues. The other 2 are muscle and nervous tissues. They're all very distinctive to look at. So there's 3 types of muscle tissue: skeletal, cardiac, and smooth. And then there's only one type of nervous tissue we're looking at. It's just regular plain old nervous tissue. The muscle tissue, the 3 kinds, we can distinguish them by whether or not they have striations, and then further based on the branching of the cells. So when I look at skeletal muscle, their striations are the little stripes. They may be a little hard to see, but those little tiger stripes are arrangements of proteins that help make the cells shorter. They produce force. That's what muscles do. So all striated muscle fibers have these striations that are regular intervals that help they contract and shorten. Skeletal muscle is also notable for being unbranched. You look, each fiber has its own connective tissue around it. They're not attached to each other. They're not electrically coupled or anything. When you compare that to cardiac muscle, you can also see the striations. A little hard to see, but they're there. They have striations. These big guys are intercalated discs. Those aren't striations. But if you look carefully, you can see that the cells are branching, that they're attaching to each other, they're breaking apart and attaching together. That helps them all attach to each other so that they're all electrically connected, so they all beat at the same time. Because that's what you want your heart to do. You want the whole heart to contract all at once. That skeletal muscle, you don't want it to do that. You want each fiber to activate or deactivate on its own so that you can control how much force your muscles are pulling with. Smooth muscle is distinctive for not having any striations. It's the only unstriated muscle that we cover. Now, it still has those fibers that shorten, but instead of being arranged in a straight line, they're wrapped around the outside. Smooth muscle can easily be mistaken for a dense, regular fibrous connective tissue, sometimes mistaken for nervous tissue or epithelial tissue. Note that it's not any of those. It does have lots of cells in the connective tissues. Those aren't cells; those are just fibers, and you can tell it's not epithelial tissue because it's not forming a boundary between one environment and another. It's just a solid wall of this muscle fiber that contracts and produces force. This is around many of your hollow organs, like your bladder and your stomach and your intestines and your aorta and so forth. Last one, nervous tissue is easily mistaken for connective tissue if you don't see these big guys. These big cells here are called neurons. Those are the cells that actually produce the electrical signaling that constitutes our thinking and our touch and our movement. All of that signals that go through our body are produced by nervous tissue, by those neurons. So if we ever give you a nervous tissue, we have to make sure we find a neuron to focus in on these big guys, because obviously this other stuff here just kind of looks like loose areolar connective tissue. But it's not. It's a nervous tissue. These other little cells are called glial cells, and they are protecting and supporting that nervous tissue, those neurons. Okay, so that's kind of a brief rundown. Make sure you study in your lab manual these different tissues, and because you are responsible for being able to identify them on site, and hopefully this has given you an idea of how to understand what they look like. And please send your lab instructor, whether it's me or someone else, any questions if you have them. All right, thanks, and enjoy, and I'll see you again soon.